Using a microscope is an essential skill to be learned in the biology laboratory. Much time can be saved in learning how to systematically find the subject of interest and get it into focus. And while the steps can be memorized, it is only through using a microscope that you can become truly proficient. First, the microscope must be placed onto the work surface. These microscopes are stored in cabinets under the bench and should be lifted using 2 hands to ensure it is not dropped or bumped. One hand should hold the arm of the microscope while the other holds the base. After plugging in the microscope, the lenses and light path are cleaned with a lint-free and non-scratching lens paper. The microscope is ready to be turned on to maximum light intensity, and the eyepieces are adjusted for optimum width and to compensate for eyes of different strengths. With the eyepieces ready, the stage is brought up to the highest position using the coarse focus knob, and the lowest power objective is selected. This microscope can hold 4 objectives, but only 3 are currently in use. This microscope has a 10, 40, and 100 power objective, although other magnifications may also be used. The slide is placed into the retaining clip, and the subject of interest is placed into the circle of illumination. While looking in the eyepieces, the outer coarse focus knob is used to lower the stage until the subject comes into focus. More precise adjustments are then done using the inner, fine focus knob. Depending on the sample, the condenser, which is the lens located below the stage, is lowered or raised to increase contrast in the image, and the diaphragm attached to the condenser can be opened or closed to select the light intensity. To pan around the slide while making observations, the vertical knobs are used to move the slide up and down, left and right. When using the 100 power oil immersion objective, the lenses are moved out of place and a drop of immersion oil is placed in the middle of the sample. Note the ring around the 100 power lens to indicate it is safe to use with oil. The 100 power objective is now rotated into place and you should see the oil make contact with the lens. A fine focus adjustment is all that should be necessary if the slide was in focus at lower power. To assist finding the focal point, some people find it easier to lightly wiggle the slide left and right, as a moving subject is easier to pick out, even when out of focus. When looking at samples under the microscope, it is important to select an area that gives the easiest to understand view. Panning around may allow you to find cells that are isolated from a group to make better observations of size or shape than when cells are stacked on top of one another. When looking at bacterial samples, it is important to not only look at the shape of the individual cells, but also the arrangement or grouping of cells. Some typical arrangements are shown here. Additionally, staining methods can be used to show different cell properties, such as the difference in bacterial cell walls with a Gram stain. Here, a Gram-positive cell is observed to have a purple color, while a Gram-negative cell is pink or red color. After making observations, the oil immersion lens must be wiped clean from the oil, and the other objectives should also be wiped. Make sure that the slide is removed from the microscope and return the microscope to its storage location.